Hello, my name is Zachary McLean, and today we're going to be talking about prokaryotic gene regulation. So first off, what is gene regulation? It's basically where a bacteria inhibits or promotes the production of a gene, and this can be due to an outside substance, such as the presence of a nutrient. So think of this like in the lac operon, which is the operon that produces the enzyme needed to break down the sugar lactose. This is in response to whether lactose is present in the body or not. So today we are going to be talking about a type of gene regulation called positive repressible. So first off, some basic anatomy. This right here is a strand of DNA. And so the operon is made up of the promoter, the operator, and then these three structural genes that will eventually be made because of this DNA. So upstream of this, we have a regulator gene. In this case, it's an activator gene. So basically, this is just a gene that either makes an activator or a repressor. In this case, it makes an activator because this is a positive type of gene regulation. And so for basic transcription, you have an RNA polymerase that binds to this promoter region, which is just a string of DNA that the RNA polymerase recognizes, and then it goes down this string of DNA to eventually make these structural genes. But there's something in between them called the operator, and you can think of this like an on and off switch for transcription. And so basically, depending on this activator molecule down here, and then you can see this little effector molecule that we'll get into in a second, depends on if transcription actually occurs or not. And so in this case, for positive repressible, this activator will then bind to this operator and allow transcription to occur. It turns it on. And so now we're going to talk about what happens if the effector molecule binds with the activator at the activator binding site. And so you can see here that this, I kind of color coordinated it that way you could tell that the blue effector molecule goes to the yellow activator and makes it green. It changes the shape of the activator. That way it cannot bind to the operator. And in that case, RNA polymerase cannot, it can bind the promoter, but then it can't transcribe these genes because transcription is turned off because this activator isn't there. So for an example, we have imagine people going on a roller coaster, but some of these people have a fear of heights that doesn't allow them to ride on the roller coaster as an example of how you can think about this. So riding the roller coaster is transcription of the structural genes. The roller coaster is the activator because you have to have the roller coaster to ride the roller coaster. And then we have the fear of heights being the effector molecule because it changes the people so that they can't ride the roller coaster. And so here are my citations.